Hi again. So now we're going to look at the final part of the genetics of Alzheimer's and Parkinson's disease lecture. Sorry. I can't speak. Lecturing takes a lot of... All right, where are we? Okay. Let's go. So another one of the genes that causes that mutations in can cause Parkinson's disease is called DJ1. It's also known as Park7, and it is also autosomal recessive Parkinson's disease, which again means both copies of the gene are mutated, and that causes Parkinson's disease. This is a less common cause of early onset Parkinson's disease. People with DJ1 mutations, both copies of their gene mutated, actually show a very classical Parkinson's disease presentation in the brain and during their life. DJ1, it's not absolutely sure what the protein is, but it appears to be a cellular sensor of oxidative stress. And it may protect neurons from oxidative stress, and it may play a role in mitochondrial function. So I've just got these two figures here. I'm not going to go through them in detail, but here's one that's talking about, what DJ1 might be doing. It might be normally inhibiting apoptotic pathways, and it might normally be activating survival pathways, and it might be activating antioxidant defense. And so this is why we say it might be protecting neurons from oxidative stress. It keeps them from dying, helps them survive, and it seems to increase antioxidants, which go out and decrease oxidative factors. So the idea might be that in familial cases, so cases where people have a mutation in the two mutations in the DJ1 gene, you get mutated DJ1, which causes inactive DJ1. But actually, like we were talking about with PINK1 and Parkin, in sporadic cases, it does actually show that oxidative stress can itself inactivate DJ1 as well. And so once it's inactivated, then you get this vicious cycle of decreased active protein and more oxidative stress. So another of the main proteins another of the main genes that, when mutated, can cause Parkinson's disease is LRK2. It stands for leucine-rich repeat kinase 2. It's also known as Park8, and it also is an autosomal dominant form of Parkinson's disease. So that means only one copy needs to be mutant before it has the effect. And so LRK2 is actually the single most common cause of inherited Parkinson's disease. It's a protein within the Rocco protein family. Again, it is late onset, so that's unusual for the familial Parkinson's disease. Usually the familial ones are earlier onset because the mutation causes the effects to happen earlier, but it's actually late onset, and its symptoms are very much like sporadic Parkinson's disease. The protein itself has a kinase activity, so if you remember, kinases are the proteins that phosphorylate, add phosphate groups to other proteins, and the reason that it's autosomal dominant, the reason that you just need one mutant copy to cause the disease, is because the mutations actually increase kinase activity. Not all of them, but that's the theory of how mutations in this gene have effects. It's a bit like if you know about oncogenes. It's the activated version that actually causes the disease versus tumor suppressors, where it's the deactivated, the inactivated, both copies needing to cause the defect in cancer. So in this case, the autosomal dominant form, it's one copy of the LRK2 protein actually has a mutation that causes more phosphorylation to happen. So it's overactivated. And so, as you can imagine, it's a kinase. There are molecules that can work as kinase inhibitors. And so for this one, it's like, there's too much activity of this kinase. That's a really druggable target. So that's something that you can really make drugs for. So the important thing is to get drugs that are specific to block this particular kinase, LRK2. That's one of the problems with inhibitors, is if they inhibit loads of things, they might have very negative effects. But at the moment, there has been some research that have shown that LRK2 kinase inhibitors are actually protective in model systems. So I think there's been some cell culture work, some animal model work, and it's being thought of as a future therapeutic option. So what happens? So it's thought that LRK2, the kinase, is involved in microtubule dynamics, immune system, autophagy, vesicles. So these would be holding neurotransmitters in the neurons, mitochondrial function, UPS alterations. This is the proteasomal degradation pathway. And possibly LRK2 actually phosphorylates alpha-synuclein as well. So what about alpha-synuclein and LRK2? There are some different theories about what's going on there. So one possibility is that alpha-synuclein directly alters LRK2 activity and that LRK2 phosphorylates alpha-synuclein. There could be an indirect interaction. They interact through one or more partner proteins. It could be that they just have sort of effects on different levels of the same pathway. So alpha-synuclein affects lysosomes, and LRK2 also affects lysosomes, but through a protein called RAB7. And it could be that they just act independently on different pathways. So essentially what they're saying is that, yes, LRK2 could phosphorylate alpha-synuclein, but it's unclear that this is actually what happens pathologically. So as I've said, like AD, there are a lot of theories of what's happening in Parkinson's disease. So one of them is oxidative stress. That's one of the biggest ones. That is what people think mostly would be causing Parkinson's disease. And part of this is because the effect is in dopaminergic neurons, and dopaminergic neurons, the dopamine synthesis pathway particularly forms sort of oxidative reactive oxygen species and things. So there's a lot of oxidative stress in dopaminergic neurons because of the fact that they're dopaminergic. And that's one of the things that is kind of special about them, and that might be why they are the ones that are most highly affected in the disease. So, whatever is happening will happen to the whole brain, but dopaminergic neurons are somehow maybe just more susceptible for that reason. And oxidative stress is something that dopaminergic neurons might have more of in general. Aggregation of pathological proteins, obviously alpha-synuclein and other proteins aggregate, decreased performance of the ubiquitin proteasome system. You saw a lot of the genes that I went through could add to that theory. A lot of them add to the oxidative stress theory. Obviously, alpha-synuclein, be one of them, adds to the aggregation theory. DJ1 was involved in apoptosis. A number of the genes, including Parkin and PINK1, might be involved in autophagy or specifically mitophagy, energy disturbances in the cell, and neurotoxic substances. So there are a lot of different things that, remember we said that Parkinson's disease is thought to be quite environmentally caused. So, just maybe pesticides or other chemicals in the environment. They might not immediately cause Parkinson's disease, or they might, but mainly for the sporadic Parkinson's disease, they might be just like adding just that little bit of extra risk that tips us over into getting Parkinson's disease. So I haven't yet talked about the genome-wide association studies for PD in any detail. So this is an indication of all the different human chromosomes. So we've got 22, and these are the locations of the the brownish ones are the autosomal dominant PD genes. So SNCA, LRK2, both of which I talked about, and VPS35. So chromosome 4, chromosome 12, chromosome 16. And then autosomal recessive PD are the orange ones. So we talked about PINK1 and DJ1, both on chromosome 1, PARK2, which is Parkin on chromosome 6, and then these other ones that are on chromosome 21 and 22. So then, the genome-wide association studies have found all of these other genes that might be important, and those ones are in sort of the coding region. And then there are these other studies that have actually looked at expression of particular genes and tried to associate that with Parkinson's disease, and those are indicated in these bluish boxes. And so this doesn't really tell you much, except that there are a lot of genes that might be involved in Parkinson's disease. So where do they fit? So there have been studies that have tried to organize, like I talked about in the data for Alzheimer's disease, like to try to fit in all these genes that have been identified in these genome-wide association studies and try to put them in different pathways. And so these yellow boxes are indications of the pathways that seem to be that a lot of the genes identified in the GWAS studies actually are in these pathways, essentially. So there are a lot that are involved in signal transduction. There are a lot that are involved in metabolism, a lot involved in vesicle-mediated transport and membrane trafficking, a lot that are involved in programmed cell death, a lot that are involved in neuronal transmission across chemical synapses, and a lot that are involved in the immune system. And then these blue boxes are also areas that, they might be involved in. And, the protein misfolding and aggregation, oxidative stress, apoptosis, lipid metabolism, endocytosis, post-translational modifications in protein metabolism, and lipid metabolism. So these are all things that a lot of them we've talked about before with individual protein mutations, individual gene mutations. So again, there are a lot of theories of Parkinson's disease, and one that that's not actually on this, except it kind of fits into the oxidative stress theory, is a mitochondrial theory of Parkinson's disease. And there are a number of bits of evidence that add to that. And so mitochondrial dysfunction is something that could cause Parkinson's disease and how. And then they've associated a lot of the single gene mutation causes of Parkinson's disease with a bunch of these different mitochondrial factors. So, as I said, oxidative stress kind of fits into the mitochondrial dysfunction because mitochondria are one of the main sources of reactive oxygen species in cells. So a lot of these individual genes that are thought to cause Parkinson mutations are thought to cause Parkinson's disease are associated with oxidative stress. Defective mitophagy, I said that Parkin and PINK1 are thought to be involved in that. Impaired mitochondrial trafficking, told you that microtubules are necessary and trafficking along microtubules is important for neurons and axons particularly because they're so big. So these proteins, LRK2, Parkin, and PINK1, might be involved in this. The electron transport chain, which is in the mitochondria, that's where ATP is produced. A lot of these mutations might be involved in that. A lot of them might be involved in altered mitochondrial dynamics, and a lot in alpha-synuclein and PINK1 in calcium imbalance and impaired biogenesis. So that was the PGC1 alpha. That's important for creating more mitochondria, which is what biogenesis means in this context. And these genes might all be involved in that. And then another theory, which I don't think was on any of the previous slides, is the iron dysregulation theory of Parkinson's disease. So it's been observed that people with Parkinson's disease have more or less iron in places where they shouldn't be. And so that's why they say iron dysregulation. It's, in a way, it's kind of iron accumulation, like there's too much, but there might be some areas where there's not enough iron. And so I was saying that dopaminergic neurons are in special, and one of the things that make them special is dopamine actually interacts with the iron pathway, and that increases oxidative stress in the cells. So again, I'm not going to go through this in detail, but essentially there's increased iron, and that has all sorts of effects on the cell, including iron overload in mitochondria, decreases in protective factors against oxidative stress, but the DNA damage can be caused by oxidative stress and increased reactive oxygen species, and this is all caused by just essentially having too much free iron in the dopaminergic neurons. So there we are. We're now at the point where I'm just gonna summarize. So Parkinson's disease and sorry, I was timing myself and I was like, wait, that's completely stopped. Just to see how long that was going. Anyway, so yes, so we have reached the point at which we are going to summarize Parkinson's disease. As you can see, with both Alzheimer's disease and Parkinson's disease, it's really unclear exactly what it is that's happening in the neurons that's causing the neurons to die. It's not clear why particular neurons die, and it's not clear what happens in the neurons to cause the neurons to die. It's known, the pathophysiology is well known, and there are correlates, like this is happening here, this is happening there, and those kinds of things are being studied, and there's a lot of information there, but it's still a big question. exactly how certain neurons are dying in these neurodegenerative diseases and exactly why they're dying in a cell signaling, within the cell, like what's happening to cause the cells to die. So, and I guess the reason I want to say that is both the Alzheimer's disease summary and the Parkinson's disease summary are very general and might not be particularly exactly what is happening in patients, because this is all an area of intense study right now, and for Parkinson's disease, it's something that I'm studying in particular. And, it's a current area of research, and it's really not exactly known, which is why, you've seen me go through all these different theories. I mean, I didn't go through any of them in detail. That would take days. But also, just to have this understanding that this is a real, for both of these diseases, exactly what's going on in this context is really not known, and it's an active area of study. And I think that's really exciting, and that's why I study it. All right, but just to give a summary of what we kind of do know, it seems that environmental toxins, neurotoxins, can cause mitochondrial dysfunction. Genetic mutations can cause these proteins not to be there. This can cause mitochondrial dysfunction and increases in reactive oxygen species, which might themselves then cause apoptosis. It's unclear. Parkin may be ubiquitinating alpha-synuclein to cause it to be degraded by the proteasome. LRK and PINK1 might be affecting the aggregation of Lewy bodies. They might be phosphorylating alpha-synuclein. Alpha-synuclein itself can be increased, and that can cause defects, the misfolding, and the Lewy bodies. So clearly, already from what I've told you in this part of the lecture, you can see that this summary diagram is not really very complete, and it's not really very accurate necessarily. So I think my point is about these kinds of aspects of exactly what causes the neurons to die in Alzheimer's disease and Parkinson's disease. It's really not known. There are a lot of theories, and there's a lot of very active research going on in these areas. So, I mean, I could have just given you an overall summary of, like, this is what happens in Alzheimer's disease. This is what happens in Parkinson's disease. And people do that, in as far as simplification to teach. And that's all good, but I think at the master's level, it's important to understand that a lot of these theories and a lot of these concepts that are taught are really huge simplifications of what's actually going on. So I hope that I've given you a flavor for the genetics of Alzheimer's and Parkinson's disease, and some of the molecular biology, and some of the signaling, and some of the details of what might be going on in the neurons of patients. And if you, if you're really interested in any of the particular concepts and you want to find out more, please do follow the links. And they're mostly review articles, just because I wasn't trying to tell you details about particular things. I needed to, even at this level, a lot of what I've told you is summary, isn't it? So, so please have a look at those if you're interested, and please contact me, if you have any questions or you can't access a particular reference and you'd like me to send it to you. So, it's too bad that I couldn't see you in person, but thank you for listening, and I hope you found this very interesting and educational. And I will hopefully be seeing you in the seminar. Bye. Thank you.